Hello everybody, my name is Jiansong Fang. I'm a professor from Guangzhou University of Chinese Medicine in China. It is my honor to give a presentation here. Many thanks to Science Father that gave me this chance. Today I'm going to introduce the recent study of me. The title is Endophenotype-based discovery and population-based validation of stem in reducing the incidence of Alzheimer's disease. Here, we look at the background. According to the World Alzheimer's report in 2020, there were 46.8 million dementia patients in 2015, and the worldwide cost is 480 billion. Overall, AD is a big challenge in the world. Nowadays, there are emerging challenges in AD drug discovery. The attraction rate for AD clinical trial is over 99%. Currently, no any small molecular drug have been approved by FDA in the past 20 years. Drug repurposing is to identify new use for approval of investigational drugs. Compared with traditional drug discovery process, drug repurposing can reduce almost 10 years to develop a new drug. It can be efficiently used to develop AD drug. So, when we look at these ongoing drugs in detail, we found that over 30 drugs are existing approved drugs. That means drug repurposing shows great promise in AD. Now you introduce some background of network medicine. A disease phenotype reflects various pathobiological processes that interact in a complex network. Disease genes have a high propensity to interact with each other, forming disease modules. The identification of this disease module can help us to identify disease pathways and predict other disease genes. In this, only about 10% of human genes have a known disease association. Understanding disease in the complex of human interaction allows us to answer some fundamental properties of the genes that are involved in this disease. Based on the concept of disease module, recent network proximity approach can identify clinically effective drugs for specific disease by quantifying the network-based relationship between drug target and disease module in the human PPI. As we all know, since drug repurposing focuses on drugs that are already on market, hypothesis testing is possible to validate the association between drug and new indication using large-scale patient-level database. Now we introduce the concept of the endophenotype module. Different diseases often have common underlying mechanisms and share intermediate endophenotypes. With this framework, a specific disease depression is the consequence of this interplay between the relevant endophenotypes and their environment. Important examples of these such endophenotypes are inflammation, fibrosis, and their essential roles in many developing diseases. Inspired by this source, we introduce this common endophenotype hypothesis in a in-file review published in Medicinal Research Review. We summarize this endophenotype, including aminos, tau, neuroinflammation, vascular dysfunction, and so on. Actually, accumulating evidence suggests synergistic interaction between A-beta and tau protein in Alzheimer's disease. Thus, we aim to develop an endophenotype module to mimic A-beta and tau interaction. We hope that this module can characterize AD as a census for efficiency therapeutic development. Then we propose a lines of endophenotype network-based framework. This framework includes construction of and validation of disease model for AD in the human interaction. In silico drug repurposing by network proximity analysis, population validation to test the drug users' relationship with AD outcomes, network-based mechanistic observation in migrate cells and iPS cells. Witness turn to test the efficiency of endophenotype module. We found that drug modules that target both amino and tau have significantly closer network distance with AD network modules built from transcriptome and proteomics data from AD genetic mouse. We collect AD staging from different sources for developing endophenotype module for AD, including 54 stagings related to amino, 27 staging relating to tau, 47 lack on SEC-AD common with genes identified by large-scale genetic study, 35 genes with at least 2 AD causing mutations from the human gene mutation database, and 36 gene corrected in at least 2 databases. Finally, we obtained 144 ADC genes. The generated ADC module includes 227 protein interactions, connected 102 unique proteins. We found all of the 144 ADC genes. 102 moved to be significantly connected through the human interaction to form a significant subnetwork. After drug screening, network proximity identified 66 drug candidates. And these drug candidates, all of them, and 40 belong to the cardiovascular system drugs, and 11 out of 40 are currently reported in clinical trials or preclinical trials for AD. This suggests the high accuracy of our approach. To test the model performance, we prioritized the same 100 candidate drugs with the lowest Z-score by ADC module using a previous promising pathway in which analysis approach, and found our approach slightly outperformed PXEA approach. Witness test the performance by an external test set. We collect 120 FDA-approved drugs with report clinical and preclinical evidence relevant. We found the network predict drug candidates are significantly enriched in the 120 collected drugs with no preclinical and clinical evidence related to AD. Applying a subject matter extends career results in Stanford as the best candidate, which has significant close distance with AD disease module. Since many clinical evidence have shown the positive effect of Stanford in AD, we stack Stanford to test the drug users' relationship with AD using population-based observation studies. We analyzed the Stanford user relationship with AD outcomes by analyzing 7.23 million patient data from market-scale medical claims. We conduct 3 types of drug cohort-based observational studies. This table refers to the description of patient data for pharmacokinetic analysis. Then we extract the KM curve conducted for propensity score strength log rank text and across a course model. Population-based analysis support the potential utilities of Stanford in reducing the risk of AD. We also perform the disease comorbidities as adjunct analysis for sildenafil. Taken together, sildenafil user was associated with a reduced AD risk in both the general population and subgroup analysis. After that, we test in vitro observation of sildenafil observation of action. Both network analysis highlighting the involved mechanism of action for sildenafil in AD and shows that CDK5 and GSK-SUN beta have closer network distance to sildenafil target than other CGN in the human interaction. We therefore validate the positive effect of sildenafil on LPS-induced activation of GSK-SUN beta and 3 betas and CDK5s in the human macular HMC3 cells. To further test the effects of sildenafil on AD, we test the effects of sildenafil in iPSC-derived neurons from patients with AD, and we found significantly increased neuron-wise growth and decreased photovoltaic tau expression in neuron models. Summarize, we give a process of concept of AD endophenotype disease modules, and the drug repositories identify 66 drug candidates. Sildenafil reduced a significant decreased risk of AD using the retrospective case control validation. And finally, we used the in vivo observation of sildenafil's mechanism of action and validate their mechanism. The results of this project have been published in Nature Engine as first author of me. This research has been highlighted by National Institute of Health. In addition, Nature Engine published a commentary titled "A Needle for Alzheimer's Disease" in a stack of claims data. Meanwhile, this paper also have high article metrics score. 304 news outlets introduced this finding, and is the top of the list of the Nature Engine papers checked by MHG. It also recommended by faculty opinion. Finally, I want to acknowledge some people, including Professor Fei-Xiong Chen. Finally, with many thanks to the scientific father international research work on network protocols. That's all. Thanks.